Can everybody see and hear me? Good? You
Yep.
can see the presentation? Okay, cool. Thanks, Michelle. So, hey, I'm Michael. I'm a PhD student at Stony Brook University. And today we're going to talk about satellites and seals. So I hope you're excited about it, because I am. So Antarctic pack ice seals, they're true seals. They're basically pretty cool furry animals that you can find basically anywhere around sort of coastal Antarctica, pretty much wherever there's some form of sea ice. Sea ice is really important for seals for a number of different reasons, not limited to sort of breeding or predator avoidance, sleeping, maybe checking out great Antarctic sunset. And there's a long history of studying seals, across the continent. A lot of great work has been done, but there's inherent challenges, for doing continent-wide surveys, right? Antarctica makes things logistically challenging for a number of reasons. So, running censuses from ships or just aerial surveys is sort of limited when you're trying to look at the entire picture of things. For years now, since the mid 2000s, we've been using satellite imagery more and more to find seals, sort of study their patterns and habitat associations, et cetera. If you look at the panel on the right, you'll see, sort of just as an example, you'll see a typical image of sea ice or pack ice. And then if we zoom in closely, we can actually see individual seals. So this is pretty cool. And this is sort of the direction where at least this project is going. The motivating ideas here are, identifying individual seal species just from satellite is basically impossible with the naked eye. But maybe we can use data fusion approaches and some cool statistical modeling to maybe find out a little bit more about what's going on, and maybe, hopefully, ultimately determine, to what degree can we discriminate individual species, by looking at things like fine scale spatial patterns. So pack ice seals, there's 4 different species of general interest in Antarctica. This study focuses on 2 of them: the Weddell seal on the top left, and then crab eaters on the lower left. Basically because they're the most abundant, and maybe so these are the target species for the scope of this project. Just some general info, for their breeding season, there's quite a bit of overlap between the 2 species, running somewhere between, September and December. Weddell seals have an overall estimate of lower abundance, but the key feature about these individuals are their sort of fast ice obligates. And so they use fast ice during the breeding season, and fast ice being basically sea ice that is land fasted, up against the continent, where it's not free floating. Crab eater seals, on the other hand, much more abundant globally, or throughout the Southern Ocean. But they really tend to rely on pack ice during the breeding season. And so pack ice is this sort of, like the picture behind what we're looking at, it's like free floating, moves around with the ocean, so versus fast ice. So these are our 2 focus species for this project. So the question is sort of, how might we approach discriminating species, and what data can we use to even reliably find seals? For this project, we're relying on high resolution satellite imagery, specifically the satellite known as Worldview 3, which is from the Maxar, formerly Digital Globe constellations, and has a spatial resolution of about 30 centimeters, so it's pretty high resolution. The imagery data sets that we've taken, sort of the source imagery for seals, it kind of is sort of spread across a bunch of Antarctica. I think most importantly, we're really trying to target Weddell seals specifically. We're really keying in on a specific area, Erebus Bay, which is in the Ross Sea, right next to McMurdo Station. And it's basically the site of, it's an established, long studied set of haul-out locations where we, can say within a given image, we can, with a high degree of confidence that, what we're looking at is actually a Weddell seal. Crab eaters, on the other hand, to try to pull imagery sort of relating to them, it's arguably more difficult, but the scenes that we're taking are again taken from the breeding season, and are focused on pack ice. And so this is working off of the loose assumption that if we're seeing a lot of seals out on pack ice during the breeding season, there's a high likelihood that a lot of those might be crab eaters. So what do we want to do here for characterizing spatial patterns? The image sort of in the right in the background has a bunch of red dots. Those are locations of seals. And for any space where we're seeing seals like this, there's some covariates of interest that we want to check out. So this ranges from, we're interested in seal body size. So it's like, how large are individuals, group size. So how many individuals are you going to find, from one individual seal? How many other individuals are there going to be within, say, 25, 50, or 100 meters? And there's more, standard sort of metrics of nearest neighbor distance. So what is the proximity of others close to you, right? So the way we approach this, just to give you an idea, sort of cradle to grave, we'll take an image, like this one over Erebus Bay, and we'll find seals using a combination of automated approaches and then also sort of human manual annotation. So we scour all these scenes, look for all the seals. If we focus in on this area in the red circle and zoom in, we can actually see sort of this peppery shot of things on the landscape, and those are actually the seals that we're looking for. So what we do is we find all these seals and we append spatial points, so we then know like the absolute location at any given time and space for these seals. We zoom out, you go through this, you spend tons of time trying to find them in a given scene, and you kind of have an output that looks something like this, right? Where like here are all the seals we can find in a given space. From there, once we have the locations, we work to extract these spatial statistics. The first pass is running a set of simple algorithms to extract proximity distances, or group size, nearest neighbor, et cetera. And then we, pass all this data, pass things into the body size calculation. So it's a little bit more involved, so I'll kind of walk through it for you. Starting from the top, moving our way down, we have an image, worldview 3 image, just of pack ice. We know where all the seals are located, and we think we do. And so for every individual, we essentially clip out the seal and its surrounding environment, and pass that raster, that image, through either a deep learning or a random forest classifier to actually extract individual seal pixels. And by getting those pixels, we have this like rough estimate of body size or surface area for any given seal. So we can then append that back, into the data set. So every seal, we know where its location, we know where it's located, in relation to its neighbors, and we have this estimate of size. Once we have all this data, we're interested in trying to sort of identify clusters and see how things are partitioning themselves. Starting with a classic function, Ripley's K, we're using a modified version known as the L function, but I'm just going to quick walk you through it to give you an idea. Basically, the idea behind Ripley's K is to try to take point sets, data points, locations or whatnot, and try to determine, through space, whether something is showing patterns of clustering or overdispersion, or if it's just, generally, a completely random pattern. On the panels, the 3 panels up top are sort of illustrations of what those patterns look like. We have uniform, we have one that's much more clustered, and then we have an overdispersed, or, things are sort of repelling from one another. The figure on the bottom here is a classic plot you'll see when you plot Ripley's K. Crossing the axis, the 45-degree line is sort of the expected K. So anything that falls right around there is maybe just just complete spatial randomness, or it's not particularly important. But in the above and below that line is where interesting things are kind of happening. In the green, we have this sort of clustered space. So between 0 to R, this point, things are showing some clustering, and then after that, there's sort of this opposite trend that shows over dispersion. So this gives us a good idea on sort of patterns of clustering, what these things look like. To apply this to our data set, Again, we have some seals, two different scenes, two different locations, and these are the raw points. If we just sort of overlay just the points themselves and sort of do this, human visual assessment, we can see that there's, at least in, the Erebus Bay, there's, looks like a considerable amount of clustering. On the other side, this one is maybe there's probably some clustering going on, but it's harder to tell by eye. So it's useful to, use these statistical analyses and these other methods to, get a clearer picture. Again, just for reference, these are the spatial points on the left that we just saw. On the right is our outputs of Ripley's K from the, our own analysis. Crab eater seals are located on the left, what we assume to be, and then Weddell seals on the right. There are two for each one. There's sort of two ranges of space that we're applying the function to. So from 0 to 250 meters, and then a more constrained 0 to 50 meters. In both cases, both for crab eaters and Weddells, if we look at that line, that sort of 0 line, we're like, definitely we're in the positive zone. So there's clear evidence of clustering going on. But what's kind of interesting is that the patterns themselves are a bit different between crab eaters on the left and Weddells on the right. So this, gives at least is evidence of, it might be worth looking into checking out how these things play out in space on a closer level. So what we do, we've run this, and then we move into a sort of a hierarchical clustering analysis. And what this does is basically using a set of covariates, that nearest neighbor distance, group size, etc. Basically, the clustering algorithm will run through and basically try to assign individuals to one class or another class or another class. In this case, we have two classes, crab eaters or Weddells. What we see here with the red and blue points are the sort of outputs. On the left, we have a scene with fast ice, which we would assume, given the breeding season, that these are likely to be Weddells. We can see anything that's in blue is classifying as Weddell seals. And the red is sort of, it's sort of like the false flag on the outside. Likewise, in this pack ice scene, we have a similar pattern here, where most of the points by and far are being properly classified. So basically, what this suggests is, even using a few different covariates of interest, specifically, a group size metric, and then nearest neighbor distance, we can sort of do a pretty reasonable job of actually assigning individuals, from one class to another. So kind of the key takeaway here, there's caveats to the study, and it's sort of still in its preliminary stage, but it looks at least like, within this case, seal species appear to sort of be roughly differentiable, along group size axes and, nearest neighbor distances, etc. I think
30 minutes, Miguel.
What's that?
30 minutes already.
Oh yeah. I'm good to go. There's just one more slide. And basically the seal size estimator, essentially what we have is it may be inadequate due to overall spatial resolution, or might need to be improved. So just moving forward, we plan to expand the data set to add more scenes, make the estimator more robust, and then ultimately the grand objective would be to sort of try to develop a model to assign the probability of seal species membership, for individual seals. So with that, I'm happy to open it up to questions for anybody who has them. Thank you.
Thank you a lot, Michael. It's wonderful presentation. So anyone have, I think you have a question. Okay, the question from Andrea Bonilla. Michael, how do you confirm that the species is being classified accurately? That's like one of the trickiest parts. So part of this, at least for the preliminary study, really relies on using those sites in Erebus Bay, which are known Weddell seal haul-out sites. We have specific reliable hollow locations, that we're working off of. And then the rest of it is sort of, running these assumptions based on either, individual literature for locations where there are, known to be a considerable amount of pack ice seals.
So part of it is, you know, really relying on using the Weddell seal classification stuff from Erebus Bay as sort of as the baseline. Obviously, just because, you know, ground truthing is near impossible unless you can, you know, task a satellite while folks are you know, out and about in a boat, you know, surveying pack ice fields.
Okay, Michael, thank you a lot. You have more questions in the chat box, the Q&A box, sorry, the Q&A box. So you could answer them. Because you don't have time, you have to move to another talk. And very, very nice presentation, Michael. I love it, the images as well.
All right.
Okay.
Well,
yes. So you can answer the questions in the Q&A chat box. So we'll go to the next one from Renan Lima from Brazil. It's my friend. So Renan will talk about ecosystem shifts from stable isotope analysis in seals. Over to you, Renan.
Thank you, Manu. So let me go ahead and just share my screen. Okay. So is it my screen showing okay?
Yes. Okay, Renan.
Okay. So I'm just gonna share with you some results of my PhD, which I've been conducting at the Federal University of Rio Grande in southern Brazil. And okay. So we know that the Southern Ocean has been changing rapidly over the last century, and the increased ocean warming events have been changing, for example, the phytoplankton community structure around the Antarctic Peninsula, and the concurrent decrease in the sea ice extent has been negatively affecting krill abundance. And future climate projections suggest that there will be a further reduction in krill abundance, and krill specialists predators will either have to switch to farther offshore or more southern foraging habitats, which will increase their energetic demands, or they'll have to change their diet, which was already simulated for predators like the crab-eater seal, for example. So that we can expect some trophic cascade effects from these environmental changes that have been occurring and that will likely still continue to occur. So the Antarctic fur seal is an important krill consumer, and this species breeds north and south of the polar front, and the numbers of the species have been increasing as they recover from the overharvesting that happened during the 19th century. And it is considered an ecosystem indicator by Camelar, since the proportion of fish to krill in their diet, as some of the biggest of their populations, seems to be related to krill availability in the environment. So in years of lower krill availability, they compensated by incorporating more fish into their diets. So, and the species may also complement their diets with other food items as well, such as penguins and cephalopods. But most of the global Antarctic fur seal population evolved in a context of high krill abundance, and so they could be less fit for consuming other prey. Detecting the influence of climate change on the ecology of elusive and long-living predators such as marine mammals and seabird is a major analytical challenge, since a long time series are often required, and we can always have data to confirm this kind of trends. So marine mammal teeth are accretionary and metabolically inert bioarchives that can kind of overcome this challenge, since they represent individual and natural chronological record of assimilated diet through the deposition of annual growth layer groups, pretty much like tree rings. So this pattern of tissue deposition allows us to construct isotopic time series. And for those that are unfamiliar with stable isotopes, these are chemical tracers that represent the diet that is truly assimilated into the predator's tissues. So they are transferred from prey to predator in a predictable way. And the most commonly used stable isotopes in ecology are carbon and nitrogen. So carbon is typically used to infer, to discriminate between different sources of primary production, and nitrogen is typically used to infer about trophic position, although it is also used to differentiate between environments, since it is also related to the different sources and sinks of nitrogen in seawater. So that way, when we are familiar with the isotope variation in an environment, we can know a lot about animal movement and its foraging habits. So we used actually teeth that were provided from a collection of Antarctic fossil skulls curated in the Marine Mammal Laboratory of the Argentine Antarctic Institute. We selected upper canines from male individuals that were collected between the 1980s and up to 2015. And those samples were collected from the South Shetland and the South Orkney Islands. And in total, we used 41 individuals. So what we basically did was, first, we estimated the age of individuals through the counting of dentin growth layer groups. And we determined the isotopic ratios of dentin samples. We obtained a portion of the dentin powder from each growth layer group using a computer-guided micromill drilling system. And the stable isotope analysis was performed using an elemental analyzer linked to an isotope ratio mass spectrometer. And then we assigned the last GLG, the last growth layer group, to the individual year of death, and back calculated calendar years for the previous GLGs. So for example, we have here a 7-year-old male that died in the year 2000. So we assigned the last GLG to the year of the individual death and back calculated years for the previous GLGs. So we have the year 2000 for the year of death, and then the previous GLG for the year 1999, then 1998, and so goes on until the first GLG that would correspond to 1994. So for each individual, we constructed an isotopic time series. So we also constructed a database of environmental variables that could potentially affect Antarctic fossil foraging, such as chlorophyll A, sea surface temperature, cryo density, and climate indexes, such as the southern annular mode and the O'Neill southern oscillation. And we modeled the relationship between all these variables and also the origin of samples, which would be South Shetland or South Orkney Islands, and carbon and nitrogen values of GLGs using generalized additive models with penalized regression splines as smoothing functions. And SAM and ENSO indexes were tested on 1, 2, and 3-year legs, considering that there are no linearities between the physical and biological processes, right? So it would be required at least 1 year for the effects of these climate anomalies to be propagated up the food web. So since chlorophyll A data was only available from 1998 onwards, we had to construct 2 sets of models for each isotope. So we constructed models from the 70s, from 1974, actually, to 2015, and another set of models from 1998 to 2015 to account for this particularity of chlorophyll A data. So first we had to look on the differences on age classes. We found no differences on nitrogen between them, but we found differences on carbon. And since subadults and adults were more similar between each other and were more, their isotopic signatures were more consistent with foraging on Antarctic waters. We focused on them to perform our temporal analysis. So we could detect a strong effect of year on the long term, considering the whole data set from the 70s to up to 2015. And this is important to emphasize that our carbon data was corrected for the Suez effect. And this temporal decrease of delta 13C was of almost 0.04 per mil per year. And there is no indication of a significant decline in phytoplankton biomass, considering the wide area that has been used by male Antarctic fur seals, which could be one of possibility to explain this trend. And it's also unlikely that the switch between the switching primary producers communities in some areas, such as the increase in cryptophytes and the decrease in diatoms, could have influenced Antarctic fur seals through direct baseline effects, since cryptophytes are a negligible prey species for krill, and the fish that are consumed by Antarctic fur seals are also krill feeding species. So what's happening here? What's explaining this decrease in carbon values over time? So the highest krill densities in the Atlantic sector of the Southern Ocean were concentrated in its northern part around South Georgia region during the 1920s and 1930s. However, since the 1970s, the distribution has contracted southward. And this latitudinal distribution shift may have actually contributed to the decreasing delta 13C values found in our data through the use of more southwardly habitats over time, which we know for a fact that are characterized by carbon depleted baseline values. So that seems more likely to have contributed to this declining trend over time in our male delta 13C values. As for delta 15N, only year showed statistical significance for the most recent period, so from the late 1990s to 2015, when it increased. So despite oscillations, there is no evidence to support a long-term decline of krill contribution to Antarctic fur seal diet, as it is still the main prey for the species foraging in Antarctic waters. And this has been corroborated by SCAT analysis by other studies as well. And we found no significant relationship between krill density and nitrogen values. So for that reason, we hypothesized that the increase in nitrogen after 1998 might actually be related to a combination of different food web shifts and also the use of different foraging areas. So for example, there's been an increase of alternative prey availability, such as gentoo penguins, who are thriving in their environment in recent years. And there's also been an increase in competition. Not only penguins
And then 12 minutes.
Okay. Not only penguins are, some penguin species are very abundant right now, such as gentoo penguins, but also some baleen whales are recovering from the hunt. And there are also krill predators. And also in the context of shifts in primary producer communities, krill might have also been forced to switch between diatoms to small zooplankton such as copepods, which would increase their trophic level, and that consequently would lead to higher nitrogen values in Antarctic fur seals. And also, we had to consider that Antarctic fur seals may also be using different areas with localized higher primary production with higher nitrogen baseline values. So the take-home message here is that individuals likely follow their main prey distribution over time, targeting areas of higher krill concentration, and increased consumption of higher trophic level prey or switch to different feeding areas after the late 1990s. However, it is important to consider here that we considered a wide geographic area in our models, which likely did not capture diverging trends in more specific regions, as the precise foraging locations of our individuals were unknown. So I'd just like to thank a few institutions that made this work possible, especially the Ecomega Laboratory and the Argentine Antarctic Institute and CNPq for the funding. So thank you very much.
Thank you, Renan. Thank you, Renan. Thank you a lot. It was a very nice presentation. So we have time for one question. Thanks, Renan. I might have missed it, but yeah. But did you adjust the delta C values for the stress effect?
Yes, yes, we did. And we still found a very declining trend.
Okay. And Hannah, just one more question for me, quick. You know, because it's nice that your results and you gave a lot of hypotheses. Do you think that which one is more suitable for you about this changing? I mean, the males could travel, you know, far away.
You mean about carbon or nitrogen?
Nitrogen.
Yeah, I think the whole food web switches and the competition, and for me it seems more likely that with lots of competitors, and it would make sense that it would pressure them to specialize, at least part of the population to specialize on alternative resources. So they wouldn't have to consume only krill, so they could have maybe pursue different resources.
Okay, thanks so much, Rana, for our presentation. It's really nice. So we're going to our, yes, thank you. For our third presentation today, it's Giselle. Yeah, Giselle. She will talk about monogamy in penguins, quite interesting. So Giselle, over you, please.
Thank you. I will share my screen. It's it?
Not yet.
Yet, a minute. And now? Yes.
Yes, yeah. Now we can see it.
Okay. Thank you very much. I would like to thank SCAR for this opportunity to show this work. My name is Gisele Dantas. I am a professor in Pontifícia Universidade Católica from Brazil. I will talk about monogamy in gentle in chimp-stray penguins. The monogamy is extra-pair copulation.
Sorry, Giselle, can you just open your presentation? Yes. In the presentation view for your PowerPoint?
Yes, I open. Don't show you?
No, it's not showing. I just make bigger and better for people to look at.
Yeah, my God.
I did you.
In the now?
Yes, perfect.
Thank you. Well, I'll come back. Well, I talk about monogamy, and the mate choice is fundamental to survival and the fitness of the individuals. So, because the similar genetic parents, you increase the probability of hairy alleles shown in the offspring. So why the peoples involved to extrapopulation? The high genetic diversity is good for the fitness, and the low genetic diversity can be reduced the fitness. Although 90% of the birds are socially monogamous, and the founder 20% the species are studied genetically. For males, the explanation for the reasons for the individual involved in extrapopulation may be more obvious, because individuals that involve in extrapopulation you produce more offspring and increase the fitness. However, for females, this is not so clear, the advantages. And for this, several hypotheses has been raised. For example, ecological hypothesis that said the females involved to extrapopulation to get help if nest defense and the foods for the chickens. Another hypothesis is the good genes, and the females have indirect benefits and for increase the genetic of the offspring. And the last hypothesis is related to genetic compatibility, and the females chose the partners because of specific losses that compatibility with the males, for example, in MHC losses that relate major histocompatibility complex, that is the immune system. So based on this, we try to investigate two penguin species, the gentle and the sheep-strip. The gentle exhibits strong social monogamy, has biparental care of the eggs and the chicks, asynchronic breeding, laying in general 2 eggs. Recent evidence that it has post-fledging provides for helping the adults, and the gentle penguins receive a high fidelity, but this can change of the breed seasons and the breed locations. For sheep-strip penguins, that is the smaller penguins of species of this genus, the Pigocelis, is endemic the continent Antarctic, and exhibit strong social monogamy, biparental care of the eggs and the chicks too, asynchronous breeding, and lay 2 eggs too. Well, what you found? The first part of this work was published in Polar Biology about the gentle penguins. We sampling the gentle penguins in Elephant Island in Stinky Point during the summer 2012 and 2013, where sampling 4 families that it has 2 adults and 2 chicks. Adults were sexed by molecular genetics using the prime P2 and the P8, and the paternity was conducted using 10 microsatellites in servo software. Well, we found a good number of the alleles in these microsatellites, and a good heterozygosity observed in all microsatellites used, and tested the Hardenberg bias and the allele nulls. So you can did the analysis using all microsatellites and exclude the microsatellite that has bias, the Hardenberg. Well, for was surprised, you found the high extrapopulation rates around the 48 75% of the chicks being from extra-pair paternity, that this corresponds to 72.5% of the nests, with less than one chick being from extra-pair paternity. However, when you test with the genetic diversity increase in nests with extra-pair paternity, you don't find genetic difference. So for these markers, the microsatellites, that is a neutron losses, that is no difference genetic. And for chimp-strap penguins, we also collect in Elephant Island during 2 summers, 2002-2003 and 2003-2004. When the first summer, you simply 52 families, and the second summer, 4 families. The same way, the adults were sexed by genetic molecular, using the primaries P2 and P8, and the paternity was conducted by 10 microsatellites using 2 server software. Well, you found worst amplification of 2 microsatellites and exclude from the analysis, and keep the analysis, the paternity, using 8 microsatellites in the end. I found 2 good number of the alleles in these microsatellites and a good heterozygosity observed. Oh, sorry. For the same way, for the chimp-strap penguins, you found the high extra-pair paternity in 2 years, in the no difference significant and between their years. In the first year of the sample, you found the 16.2 points of the chicks from extra-pair paternity, that correspond to 69% of the nests, with at least 1 chick being from extra-pair paternity. In the second year, you found around 63% of the chicks being from extra-pair paternity, and that correspond to 76% of the nests with at least 1 chick from extra-pair paternity. That is very high for that was expected if found. Well, the same way, when you compare the 2 breedings, you don't find a difference significant of the genetic diversity between the nests with extra-pair paternity and without extra-pair paternity. So, we was expected, found the low rates, the extra-pair paternity due to parental carity and the low divorce rates in seabirds. However, the genetic monogamy have been found in less than 25% of the socially monogamic birds. Our results support that the gentle and the chimp-strap penguin is not strictly monogamy, and the mate behavior as previously told, including infidelity. And the main hypothesis for why female engage in extra-pair behavior that I investigated here is that increase the genetic diversity is not supported for our data. So the gentle and the chimpanzee penguin in Elephant Island show high extra-pair paternity, being around 70% of the nests. It's important to conduct this kind of studies in other locations, other breed colonies, to investigate that is relation of the climate of their specific site or not. The reasons for this behavior, extra-pair paternity in penguin colonies, is not clear, since increased genetic diversity was not observed. And the other mechanisms like ecological mechanisms and the hypothesis for sperm stocks or genetic compatibility of the immune system needed to be investigated. This work was conducted with collaboration of several people. I need to thank you for Luana, Gisele, and Luar, that was students helping in conducting laboratory analysis. The Maria Virginia Petri that introduced me in Antarctic research helped me in this study, permit me collecting the samples in the FIDO, and the Gabriela Vela and Roberta that was students for Maria Virginia that helped me in finding work. Well, I don't know, I talk so fast. Thank you very much.
Thank you very much, Gisele. No, it was wonderful, exactly on time. So you have time for questions. If anyone, please raise your hand here, or you can write as well in the Q&A box. So you don't for now. So anyone, you have time for questions? If you know, I do have one. Gisele? Yeah. So Gisele, you said that this monogamy is different among the colonies, isn't? Do you have? Do you?
Yes.
I will compare with other colonies in Antarctica.
I studied only Elephant Island, one colony in Elephant Island, but for get the sure that is a pattern or is a specific for ecological system in Elephant Island, it's not important to do in another sites because I don't have results from other colonies.
I think we have one question here for David. What about population size, Gisele? A number of studies are showing that penguins, at least there is a diet shift as populations and presumably interspecific competition increase. But I think that's not, it's David for her, that's question, sorry. I think probably it's from the one behind. Yeah, sorry, it's the question is for the past one. Okay, no more questions. Yeah, that was Renata. Because I read penguin and then
yes, okay.
Yeah, sorry, it was for Renata. Okay. No more questions now, so we can follow. Just remember, people, that when finish all the talks, you can have a more 10 or 15 minutes talking about this session, okay? So we'll move. Thank you very much, Gisele.
Thank you very much.
We move to the next one, that is Maria. Okay, Maria will talk about influenza detection in penguins.
Hello, thank you. I think I cannot start sharing screen while others are sharing. Let me see. Okay, I think now it's going. Just a second. Good morning, good evening. Nice to meet you. Thank you very much for considering our work for presentation. I'm also grateful for participating, and I'm learning a lot from other speakers' talks. And today I would like to present our preliminary studies about detection of influenza A in fecal samples of penguins. And I am from Fiocruz Foundation, from Laboratory of Respiratory Viruses and Measles. And so I'm sending all greetings from Brazil for all researchers I already had the opportunity to see other researchers that I already met during our expeditions. So it's really nice to meet again on the conference. Well, our project actually is making part of greater projects, FIONTA, that started in 2019. And who coordinates this project is Hindell. And idea of the project is to investigate bacteria, fungus, and viruses in Antarctica, in South Shetland Islands, with the support of Brazil Marine and Comandante Ferraz Antarctic Station, where you have a lab, a new opened lab since 2020. I'm also inviting you to visit the site of the project when we have all results from other groups participating in these studies. Well, our part is actually respiratory viruses. And I would like to give you some background because I didn't see any in the program other presentations about viruses or diseases. So I would like to give you some background why we are looking for those viruses in birds. Most of the new emerging diseases are zoonotic. Some come from wild animals. And 60% of infectious diseases are coming from animals. And we have billions of cases of illness and deaths on the worldwide. You can see this on like ongoing pandemic of SARS. And actually we are expecting that new pandemic will be influenza pandemic. One of the virus influenza, it's incredible. It's really tricky and amazing virus. And its genome is divided in 8 parts. And as you can see on the you have inside the envelope of the virus, we have RNA, and on the surface, we have glycoproteins called hemagglutinin and neuraminidase. And we have a lot of subtypes depending on those glycoproteins. So probably you heard about H1N1, IH5, and so on. So all those viruses are circulating in wild birds, especially aquatic birds like swans, geese, sandpipers, ducks. And most of those viruses are not pathogenic for birds. However, sometimes it's happening that those viruses are passing for domestic animals, especially poultry like chickens. And for chickens, they can pass to swine, and swine can pass those viruses for humans. And especially swines are important because swines have receptors for many types of influenza viruses. So so it causes a huge pandemic in the past, like during the First War in 1918, we have Spanish flu that caused millions of deaths. Then we have other pandemics from like Asia influenza, Hong Kong influenza, when we have other thousands, hundreds of deaths. And always it happened that from wide populations of birds, some subtypes of influenza enter to human population. Except of that, in birds populations, we have strains usually not pathogenic, but sometimes they can change and be pathogenic. And it's happening sometimes that it can cause huge mortality in birds. And in recent times, we observed increase in those cases, increase in numbers of subtypes of avian influenza, and that infect birds, increase the number of bird species that virus affects. And what about Antarctic? In Antarctic, you have huge population of penguins, and there are already a lot of studies looking for viruses in those populations. Already from the 70s, from the 80s, people started investigating using serology approach. In recent years with metagenomic, we have increase of number of new viruses described in, not in penguins, but also in mammals and other species of birds in Atlantic continent. And for example, in penguins, various families of viruses have been already described, including influenza viruses. However, most of the results we have till now are based on serology. Only few are based on molecular studies using sequencing. And what is worrying us, the instant contact between tourists and what this can cause in animal populations, and what actually we have also in the Antarctic continent and what can pass to the other continents from there. So this is like two sides of the impact. And as you can, for me, when I came first time for Antarctic, I was surprised that how much trash we can actually find on the beach, on the colonies of penguins, and how many tourists are coming, and how close they are coming to animals, how they enter into contact, very close contact with animals. And the idea of our studies was to collect fecal samples in the South Shetland archipelago. And here in the red spots, we have some collection points. We collected in the season 2019-2020, 95 avian fecal samples. And we are going to penguins colonies, usually Chinstrap, Gento, Adélie. And we are collecting exactly this type of samples, fresh fecal samples. And we are preserving this in some special medium for preserving virus. And then we are freezing those samples to take to the laboratory for future testing. In the laboratory, we're extracting RNA and first making real-time PCR to screen the samples for the presence of all influenza A. And then the positive samples we sequenced in Illumina MySeq system. We try also to isolate these viruses in chicken eggs. And this was actually not possible because although we found some samples positive, probably the quantity of the virus was not enough and we are not succeed in isolation. However, in 5 fecal samples from colonies, mainly from chinstrap penguin and from one individual of Adelie penguin. In these phases, we found positive samples. And from two of them, we are able to obtain complete genomes. All those sequences we deposit in G site, this site when we deposit all influenza viruses, and what kind of analysis we made to discover what kind of virus it is. We performed a phylogenetic analysis, and for influenza, you make those analyses for, you remember that I told you that each influenza, virus influenza is divided, has the 8 fragments. So for each fragment, you make separately a phylogenetic tree. And we discovered here that taking into consideration the fragments that of the gene that codified glycoproteins, hemagglutinin and neuraminidase, that our samples marked here in blue are clustered together with other Antarctic influenza viruses. And we make those analyses for all old fragments of influenza. We always observed, yes?
12 minutes.
Yeah, okay. And we discovered that those samples came probably from North America, about like 30-40 years ago, and they became endemic in the populations of penguins. And what kind of influenza is that? It's called HL11H2, and it has already been discovered, recorded first time in 2011, and later we had 2 other registers in in the islands, actually the region, the distance between 100 kilometers from our from the point that we found this influenza. So we can see that actually this influenza is already circulating for some years in the area. But as you can see, this is a very small region. Actually, we don't have any other information for other areas from Antarctic. And what is the question? How this influenza came to South America, from South America, probably to Antarctic Peninsula, to South Shetland Islands. For example, it could happen through the snowy sheepbear, because it migrates and spending the summer on South Shetland Islands, and the winter time is coming even to the south of Brazil. And it's how the transmission of influenza is through the oral fecal route. Probably this could be the way that penguins, in fact, get this influenza from the continent. Of course, there are other species like sharks, skuas, and gulls, and they can also contribute into spreading the influenza in the region. And just to show you, for example, how far the influenza can travel and can even come.
Sorry, Maria, it's 15 minutes. Could you try to conclude?
Yes, I said the last slide. How many subtypes of influenza you have in just in Brazil, and how easy this influenza can come even from the north to south hemisphere. And of course, it's very important for us to continue looking for other influenza, and especially those methods without catching birds, but screening species, not to stressing birds, are very important. And I would like to thank you for those groups to contribute. And if you have any future questions, here's my contacts. And once again, thank you very much.
Thank you very much, Maria. Very nice work. And sorry but we are running time. Yes, you use it all your time.
No, it's okay.
Yeah, don't worry, because people, if you have questions to Maria, please the chat, the Q&A box, and then she will answer. Okay.
Thank you.
So we'll move to the next presentation for José Xavier, my great, wonderful friend. Please, the wonderful IT team, José recorded his presentation. So
Perfect.
we can, yeah. So, but José is around to answer the question. Okay.
Thank you. I'll see you soon.
Hi, everyone. It's a pleasure to be here. This presentation brings all generally the work that we've been doing the last few years, particularly on albatrosses and seals, in understanding the role of Southern Ocean cephalopods. Cephalopods are talking about squid and octopods that from intermediate role within the marine food webs is well acknowledged. And this is the work that we've been doing for our numerous colleagues around the world. And we'd like to acknowledge, or I'd like to acknowledge particularly my team. This is the last time we were together last week on the beach. Yes, I think it's quite important to recognize the work of our team, and not only talking about work in the lab or in the field, but also on the beach. Why not? This is a recognition of the excellent work they've been doing all through these years together in understanding how the Southern Ocean actually functions. In understanding how the organisms can be bioindicators, there's numerous papers about it. But one of the key issues that is relevant is that when we're talking about top predators, they are essential to understand how actually Southern Ocean functions. But also they can be used as a major tool to also understanding how the Southern Ocean actually functions, but also how it relates to these threats, but also how we can also use them as major vehicles to conservation. And there's a lot of work on it, but I'll give some examples of what we've been doing. And when we're talking about climate change, we're talking about major issues. We're talking about that generally most of the countries are not fulfilling their objectives in fulfilling the Paris Agreement. And there's a lot of work that needs to be done. From the Southern Ocean perspective, we need to be united and provide the evidence. This was very nicely identified at the United Nations Oceans Conference, and recognizing the relevance of the oceans to the rest, to the world, to the countries. But there's a lot of work to be done. And this was already nicely portrayed by Stephen and Cassandra when we were talking about the Southern Ocean, because the impact of Antarctica to the rest of the planet is considerably high. And the IPCC reports highly acknowledges the relevance of Southern Ocean to the planet and its consequences. But the pace of change is so high, the changes that are occurring in the Southern Ocean is so high, but in terms of implementing policies is quite more challenging. We've been doing our job. We had a workshop in Portugal that in 2021 there was this review led by Julian that emphasized some of the challenges that we have regarding the Antarctic and the Southern Ocean. So it's an updated, but also how the connectivity of the Southern Ocean to other parts of the oceans is quite important. This is an example of the humpback whales, about their migrations, annual migrations from and to the Southern Ocean are quite relevant in, for example, possibly in the future in becoming vehicles for invasive species, for example. But this is just an example of possibilities. But this connectivity to the rest of the world is something that needs to be reinforced and recognized, and more needs to be done. And when we're talking about cephalopods, what is nicely emphasized is there's no data when talking about historical impacts and what is driving these impacts. And we might take some advantage. Squid might adapt well to climate change, but there's still a lot of unknowns. But knowing that historically there's no data is something that we tackled recently, and this is what we're going to show you. So this is some of the work led really nicely by Gilda. The one point I'd like to emphasize is also that even the basic information about distribution, classification, identification is still many challenges. And we've been contributing under the flag of SCAR, most recently on crustaceans, but also on cephalopods. And this is work that I think we need to keep on pushing, not just cutting edge science, but just the basic biology needs to be addressed. And there's still a lot of gaps that needs to, then we need some answers to actually provide good guidance for developmental policies, or just understanding their biology. If we're talking about seals, this work led by Jose was very nicely, because it showed that in terms of seals, when there's no krill available, which is these black bars, when there's not much krill around, they feed more on squid offshore. And when there's more krill around in years, and there's more krill around, they actually stay more onshore. These dynamics are very interesting, although it's a small time scale, but this behavior on looking at the prey and the predator interactions, how they deal with food availability, is one of something that we need to address in the future for newest predators across the Southern Ocean. But also looking at nearby in this sense of connectivity on work led by numerous colleagues from Gustavo Daneri in Argentina on how South American sea lions can be really nice bioindicators on the cephalopod biology and fauna in a certain region. From a long-term perspective, one of the major breakthroughs that we had recently is we finally got access to data from the last nearly 50 years, since 1976 to 2016, and look at some of the most important species in Southern Ocean predators. And so this is the first time we had an organism, a nectonic species from the Southern Ocean, in understanding how they changed in such a long-term period. And what they've shown is that generally they have a tendency to species to go to a more warmer waters, as you can see here. So the carbon actually reduces some of the species. Other species have little changes, and others, for example, have a tendency to go to more colder waters. But in general term, the changes of carbon, the general trend was that they do try to adapt to changes in the environment. So they change their temperature, their adaptation to changes in temperature. So they're able to do it, but their trophic level didn't change. So they do change their habitat preferences, but in terms of their diets, they didn't change. So they are able to adapt. So it gives us the first idea that these squid might be able to cope with some level of change. So they are able to adapt in terms of their adaptation to habitat, but their trophic levels maintains the same. When we look at their relevance in terms of availability to predators. This is an example for gray-headed and black-browed albatrosses. It shows in a period from 96 to 2016, it shows that, for example, the relevance of cephalopods maintains this, it goes down a little bit, but generally stays very constant. So nearly half of the diet of gray albatrosses maintains constantly, more or less, although there's some years it goes really down, but generally gets fairly half of the diet is relatively constant. Whereas for black browse, although in lower levels, but the trend is the same. So what it means that from a longer term perspective, we have a tendency for cephalopods to be a reliable source of prey. I should also emphasize the job of PhD students that are already doing a lot of work, not necessarily from a long-term perspective, but recognizing also other predators like penguins or Antarctic toothfish that are commercially important species for the Southern Ocean. Their work is essential to development and learning more about the role of cephalopods into the Southern Ocean ecosystems. So a really big thank to them. Also recognizing the relevance of the Southern Ocean Action Plan that brings us not only the challenges that we have for the Southern Ocean, but also actions that we should take in the future in interaction with other United Nations decade actions on oceans elsewhere. A final word of remembering some key people within SCAR. We're already in 2022, but very, very important people within our community are disappearing, and we miss them a lot. So thank you very much, everyone. I hope you're having a good SCAR conference. All the best.
Thank you very much, José. Yes.
Yeah,
So
thank you.
please, thank you. So José is here. If you have any questions, please or raise your hand or you can write in the box. But please take advantage because José is someone that has a great knowledge about cephalopods and Southern Ocean.
It's a pleasure. Yeah, thank you, Manu, and for the opportunity. You could see that I've been caught in the middle of vacations, so but it's a pleasure to be all of you. I still miss all of you in person, but I think the SCAR conference has been very interesting. From our work, I should reinforce the work that the team has been doing. COVID situation was not very easy everywhere in the world. But I think we are, from the role of cephalopods in the Southern Ocean and working with the wide range of predators, we advancing solidly with the work. One aspect that I haven't touched, and possibly we need to address that more in the future, is keep reinforcing the international cooperation. I think we had really nice presentations today, and in our AgBAM group, birds and marine mammals group. But these interactions in this case, from this perspective, from the prey perspective, I think it adds another extra layer to what we've been doing. And so we really thank and having this opportunity to use your trop predators to understand what the prey has been doing and the potential impacts in the future. One of the things that is a major answer provided in these studies is that probably cephalopods could be one of the winners. The squid could be one of the winners from climate change changes. Although there's some questions in terms of abundance, but we have no evidence from the predators that actually their numbers are decreasing. Actually, they are adapting.
No, it's a very good point. And what I'd like to ask you is about that. How did, because all your works, you do a lot of your students and you published a lot. What about now, more or less, what you think about the plasticity of cephalopods? You think that they can have a better?
It's a good question. We're addressing that from the crustaceans, from the fish, from cephalopods, is putting genomics into this, is to using new techniques, DNA, genomics, transcriptomics, understanding really what is driving their adaptability. And what are the tipping points, how that transfers to the abundance of these organisms, and what will be the impacts in the food web ecosystems across the Southern Ocean.
we have a really good data from the top predators that has been already expressed through the decades. We need to go back to our funding agencies and say that top predators continue and should be enforced to have long-term funding to do this research, and particularly looking at these interactions, food web interactions, where they feed, what they feed, and how they're likely to adapt, and how we protect it. There's these amazing workshops starting on Monday. Of course, AgBAM has been leading this by Cassandra and the team. There was a presentation already on this, and we totally asked to get involved. This is very inspiring, and SCAR is leading the science, and that's what we need. From the cephalopods, we're going in another direction. They are probably some of the winners, but there's still some big questions before saying definitely yes. But we're going in another direction that they seems to be adaptable. So
okay.
Fingers crossed.
And fingers crossed. And Jose, I have one question for you from David. Jose, squid are eating machines. So there is an irony in referring to predators versus squid.
That's a very good point, my friend. Thank you, David. It's a very good point. We always see them as prey of the top predators, being an AgBAM forum. But yes, it's true. There's big questions also about on their diets. We do a lot on stable isotopes, again, on the squid now. And we continue to trying to lead some of this work from a long-term perspective, but looking at their physiology. Now we have a new project looking at these genomics, transcriptomics, trying to really understand, do they have the genes to adapt, and their diets as well. There's still a lot of work to do. One thing that we see from the food web modeling perspective, squid is still in one big box. We need to break that into leading species. There's Moritotopsis longimana, we have Mesocoteuthis hamiltoni. We have like major squid that play a key role on numerous predators. They feed a lot, it's true, but very poorly is known. It's definitely a big gap. Thank you, David.
Okay, Jose, thank you so much for your contribution. I'd like to thank you all, but you have to move on now, no more time. So if you have any more questions for Jose, please use the Q&A box. And getting this, what Jose talked about, the top predators, in order to know more about the top predators. So that is the next talk from Ryan. So Ryan will talk about predator bioregions in Southern Ocean and MPAs. So over you, Ryan.
Thanks, Manu. Let me. Here. Can you just confirm that my screen is full screen for you?
Yes, perfect.
Great, thank you. And thanks for the time slot. So today I'd like to speak about a really collaborative piece of work involving the numerous people mentioned on this slide. And I'd like to point out from the beginning that this work has just been published in Biological Conservation. So if it interests you, please do have a read of that paper linked through this QR code, and I'll also share the link in the chat. So because humans are increasingly impacting marine ecosystems or marine systems, a common tool to deal with this is marine protected areas. From 2010 to 2019, global coverage of marine protected areas increased from 2.9 to 7.5%, but this is still well short of the 10% or more ambitious 30% targets that have been proposed. If we consider only fully protected MPAs where no extractive activities are allowed, or highly protected MPAs where only light extractive activities are allowed, MPAs cover only 2.7% of our oceans. Marine protected areas should be ecologically representative to be effective. So we're not just aiming for straightforward aerial thresholds. This representativeness is interpreted in various ways, including coverage of certain species, coverage of those species niches, or coverage of ecoregions or bioregions. And bioregions is the term that I'll be using from here on out, defined as a biological and physical partitioning of geographic space based on spatial distribution of multiple species, communities, ecosystems, or any other biological characteristics. Turning our attention to the Southern Ocean, two previous regionalizations have been produced, at least two. The first one of these was a benthic regionalization by Douglas and colleagues, and that was based on biological data, which included species distribution and endemism, as well as existing bioregion definitions and abiotic data layers. A bit late in the same year, actually, Ben Raymond produced a pelagic regionalization based on ocean depth, sea surface temperature, and the proportion of time that a given cell was covered by sea ice. Cassandra Brooks and her colleagues assessed the representation of those two regionalizations in the current MPA network in the Southern Ocean, and found that half of the pelagic bioregions fell short of even 10% representation, and most of the benthic bioregions fell short of that 10% representation target. So there's a clear need for expanding the MPA network in the Southern Ocean. In tackling that need, our early work as part of the retrospective analysis of Antarctic tracking data used the rationale that marine top predators can indicate or signal areas of ecological significance in the Southern Ocean. So using tracking data for thousands of marine predators from 17 species, we modeled their habitat selection and combined those species-specific predictions to produce a set of, or an integrated set of predictions signaling these areas of ecological significance, which are shown in white contours on the bottom left map, and then just highlighted as blue polygons on the main map here. However, we subsequently recognized that there was an opportunity to use these predictions in more detail to examine this bioregions concept. So I'm just going to chat very simply through the analysis. You can ask me about any further details or read about them in the paper. But essentially, we used the species-specific habitat importance predictions, and we detected statistical clusters in those using the Clara algorithm. So on the left-hand side of the screen, you can see the maps for each species, which are subject to the clustering algorithm. And then on the large map with the many colors over here, we've actually mapped the statistical clusters. So each different color represents a different cluster, and I'll return to this in a moment. So again, on the map on the left, we have those statistical clusters mapped out. Let's move that. And then on the right-hand side, what's represented here is hierarchical clustering of those clusters. So we can examine the relationship among the clusters, essentially. We chose in the end 17 different clusters, but in order to make it a bit more tractable and to sort of explore the higher level relationships among those clusters, we also defined 4 what we call superclusters or high level clusters, which roughly correspond with the Antarctic. So that's clusters 13 to 17, the Scotia Arc, clusters 9 and 10, what we're calling distant subantarctic, because they're subantarctic that lie some distance from the islands, and then a set of subantarctic clusters, which lie more proximate to the islands. The heat map below the hierarchical clustering just represents the mean habitat importance of a given species in that cluster. So for example, if we look at the top row, white-chinned petrels, we can see that they have high mean habitat importance in the Scotia Arc, in the 2 Scotia Arc clusters. Or if we look at Antarctic fur seals at the bottom, for example, we can see that they also have very high Scotia Arc importance. some importance in some Antarctic clusters, and then relatively low importance or habitat importance for the subantarctic clusters. So we can also examine the environmental covariates that are driving these clusters, because in these bioregionalization exercises, it's essential to ensure their validity. That is, to ensure that we're actually capturing the processes which are driving the observed biological patterns. And so we used the environmental covariates, which went into the original habitat importance models. In the paper, there's some different representations of the environmental covariates, but over here, I'm just presenting a non-metric multi-dimensional scaling of the cluster space. So what we do is essentially to reduce the dimensionality and map the clusters onto 2 dimensions, which is shown in this panel over here. And then we can look at the correlation of the environmental covariates with that ordination. So each of these vectors on the NMDS plot represents a single environmental covariate. And the labels in the boxes over here refer to the labels on this panel over here. Now, this panel shows the correlation of each vector, which you see here, with the ordination. So it means that the higher correlation means that environmental covariate plays an important role essentially in separating out the clusters. So if we look at the most important one, CI concentration, we can see that's associated, as we expect, with the set of Antarctic clusters. Taking another example in the other direction, we can see that CI accessibility or surface wind speed is associated with clusters in the other direction. We were also very interested in what prey patterns may be underlying these clusters. So just very quickly, we obtained modeled habitat importance for several myctophid, euphorid, and cephalopod species, and used the random forest classification model to try and determine what species essentially discriminated among the clusters. And we found that the most important species discriminating across all 17 clusters was the myctophid Electrona colesbergii. Interestingly, Antarctic krill were not so influential. You can see them down here. And finally, we wanted to look at the coverage of these clusters in the existing and proposed MPA network. So if we look at panel A up over here, we can see the actual area in millions of square kilometers of each cluster, which is encompassed in designated no-take MPAs in blue, designated mixed-use MPAs in green, proposed MPAs in orange, or which lie outside of MPAs in gray. If we break that down by area percentage, and we superimpose 10% and 30% thresholds on these, we can see that quite a few of the clusters do meet the 10% threshold, but very few clusters actually meet the 30% threshold. And if we sort of look at the higher level clusters, so by broader geographic region, we can see that the distant subantarctic is very poorly represented in any kind of MPAs. The subantarctic currently meets 10% threshold, but there's very little potential to reach the 30% threshold with very few proposed MPAs. The Scotia arc can nearly reach the 30% threshold if proposed MPAs are designated. And the Antarctic currently meets the 10% threshold and can reach the 30% threshold if the proposed MPAs are designated. So in summary, marine protected areas should be ecologically representative to be effective. And we produced a Southern Ocean bioregionalization based on predator habitat use. We identified 17 clusters falling into 4 larger groups. Some of these clusters are not adequately represented in marine protected areas. However, proposed marine protected areas can contribute to meeting conservation targets. I'd like to thank CESAB FRB and WWF for funding, and all the rapid data contributors for kindly contributing their data for this work. Thank you.
Thank you, Ryan. Yes, you have time for questions, for 2 questions at least. So let's see, you can raise your hand or write on the box. Yeah, still waiting. So Ryan, as you said that there is some groups that are represented in the MPAs. Is there now any advice for the new MPAs when meetings about include these areas and animals?
So if you recall the MPA figure, the Antarctic can do quite well if the currently proposed MPAs are designated. We project that they could reach that 30% threshold. However, the sub-Antarctic is quite worrying. As you saw, there were essentially no new proposals. So there's very little potential currently to reach that 30% threshold. And the main reason for that is that a lot of those sub-Antarctic areas are actually beyond national jurisdiction. So nations on their own have very little potential to currently meet those thresholds. So that really requires international coordination as well as new legislation to provide a mechanism for declaring MPAs in the high seas if we're going to meet that threshold in the sub-Antarctic.
Yeah, because I think that's a trick. One part of MPAs are inside no territories, and another are inside territories, isn't it?
Yeah, that's exactly right. But we make the point in the paper that CAMLR has already provided a fairly successful model for international coordination in terms of managing marine ecosystems. And together with the ongoing UN negotiations on this, I wouldn't use the word that we're optimistic, but there is certainly potential for creating the mechanism to do exactly that.
Okay, so no one, any more questions? Yes. Okay, so you move on. If you have any other questions, please you can use the now.
Yeah, Teo has a question.
Yeah.
Okay, sorry. Go ahead. Yeah, there is one. I think is your question. Let's see. I don't have here. Maybe in the chat.
Okay. Hello, everyone. Can you hear me?
Yeah. Yes.
Yeah.
All right. Yeah. Thank you for the presentation. I was just wondering. So, if I understand correctly, so you take a lot of tracks of GPS tracks for a lot of species, but I was wondering when doing the clusters, do you use the whole track or only the feeding areas that you identify using some analysis?
Yeah. So I should have explained that better. Thanks for the question, Teo. Actually, it's one step even further than that. So we use the tracking data to build habitat selection models, and then we predict those models across the entire Southern Ocean. So for each species, we have a map or a layer of the probability of them selecting that cell, so from 0 to 100 actually. And then we use those maps with a layer for each species to run the clustering. So we don't distinguish foraging areas from non-foraging areas. We're simply using those values from 0 to 100 for each species and clustering them. So clusters could mean that species similarly do use that area, or species similarly avoid that area. And so in the paper's supplement, you'll see there's a figure in which we plotted the habitat importance for each species by cluster, which gives you a more nuanced idea about which species, shall we say, like each cluster and which species don't like them. If that explains it, sorry, it's a little bit complicated.
All right, no, thank you. It was very clear. Thank you for the answer.
Thank you, Ryan. Thank you, Theo. So we'll move on to our last talk from the birds and marine mammal sessions, but not least, of course. I'd like to invite Susanna. She'll talk about spatial temporal consistency of penguins. So Susanna, welcome.
Hello, everybody. I will just share this. Okay. Can you see everything in the full screen?
Yes, perfect.
Perfect. Thank you. Thank you very much. So I'm Susanna Zaikova, and today I would like to present you results of my recent postdoc project, where we wondered whether Adelie penguins care about boundaries, and in particular, we were interested in spatial temporal consistency in their wintering behavior and in the implication for the conservation. So the Southern Ocean and Antarctica are home to unique biodiversity. However, this remote ecosystem is threatened by effects of climate change, increased human pressure, invasive species, and nonetheless the fisheries. One of the ways these threats can be mitigated is through marine protected areas, which within the Southern Ocean and Antarctica are managed through the CAMLAR. Currently, there are two existing marine protected areas, one in the Ross Sea and the other one in the South Orkney Island, and three proposed protected areas in Antarctic Peninsula, Weddell Sea, and in the East Antarctic. And in this talk, I would like to focus on the East Antarctic marine protected area proposal. So this proposal was proposed, I'd say, back in 2011 for the first time, and since then it went through several changes and adjustments. And currently, this proposal consists of three areas, which is one Meg-Robertson, Drigalski, and the Orwell Seamet. One might wonder why this area and how they were delimited. When you take a look in the original proposal, we can see that the size of the area is determined by Adelie penguins' foraging requirements, particularly during the critical breeding period, also including the foraging areas of emperor penguins. So which we perfectly see in the figure on the side on the map, where the color polygons refer to the estimated foraging areas of Adelie penguins during the breeding season within the three proposed areas, which are in blue. And when you take a look closer on one of these areas, which is the Orwell Seamet, and consider only foraging trig of Adelie penguins during the breeding season, we see that they almost perfectly fit within the proposal. However, we know that the Adelie penguins, after the end of the breeding, spend their wintering areas after the breeding season, and may migrate to distant areas, which this wintering period in Adelie penguins may last up to 8 months. Therefore, we are wondering how Adelie penguins would be protected within this marine protected area proposal during their wintering period. We have already some first hints from the first year of tracking of Adelie penguins from the area in the Tech Adelie, where the authors found that the Adelie penguins in general spend really low proportion of their time during the wintering period within the proposed marine protected area, which on average is less than 30% of the year-round trips. So we aim in this work to identify wintering areas for Adelie penguins over multiple years and to assess interannual spatial consistency in the use of wintering areas. Also, we were interested to quantify the use of the proposed marine protected area Deauville CMERT. And also we are interested to infer behavioral changes of Adelie penguins during the wintering season. So we studied Adelie penguin, which is also listed as a bioindicator species of CAMLAR. We studied the individuals from Ile de Petrel in Terre Adelie. We used the geolocators to track the wintering movements over 5 years, almost consecutive 5 years from 2015 until 2020. And a quick glance over the methods used. To estimate the position, we used the probabilistic algorithm ProbGLS. We quantified activity budgets, which we used as a proxy for foraging behavior of Adelie penguins from the wet-dry sensors. We calculated kernel utilization distributions per year and used these values to infer overlap and consistency, interannual consistency in the wintering areas using Bhattacharya affinity index. And also we were interested to quantify the time, the amount of time individuals spent within the boundaries of the proposed marine protected areas. And also, we also identified the side of the potential importance for conservation through the approach developed to identify key biodiversity areas. So I will start over with the results about the activity budgets over the wintering season of Adelie penguins. In this figure, you can see on the x-axis time from the almost deployment to the recovery of the loggers. Each color reflect the mean values of the activity budgets of all tracked individuals and standard deviations. And on the y-axis, you can see the time that the animals spend in the water during the day. Also, this information, a part of serving as a proxy of the foraging behavior, it's also valuable to infer the phenology of the species, where we identified as a molting period the amount of time, the days where the individuals spend time in dry, and we use this information to infer the beginning of the wintering period until the end of the wintering period, where the animals return back to the colony. Regarding the wintering areas, over the 5 years, we observed in each map refers to each tracked year. We observed that the individuals perform migratory movement westward from the colony, where the colony is Ile de Petrel in this triangle. The movements were mostly bounded by the 60 latitude degree and the depth of 1,000 meters. And individuals reach the maximum distance from the colony between July and August, and the distance was approximately 1,600 kilometers. We saw that the individuals really restrictively use the area of proposed protected area Deauville CMERT, where only 12% of their locations were located, on average, were located within this area during the wintering period. These values were higher during the first 2 years, where animals stayed closer to the colony. However, all over the last 3 years, the values were really close and almost constant. On the other side, we observed that the individuals also reach in several years, in particular the last 3 years, the other marine protected area, Drigalski, and spend more than 25 of their individual tracks within this area. We found a high interannual overlap in between the wintering areas of Adelie penguins. When we consider overall distribution, we found the values range from 0.8 until 0.96, where the maximum value is 1, which indicates almost identical distribution. Considering only the 50% utilization distribution, where the maximum value is 0.5. Interannual overlap was also high, which the areas are delimited with the lines. So using the approach to identify a site of potential importance for conservation through the key biodiversity area approach, which is based on the individual distribution of target individuals. We identified in the red the area which is the area of a potential interest for conservation during the wintering period of Adelie penguins, which part of this area lays within the proposed protected area, the Hindell Seamert. However, majority of the area lays just in between the two proposed protected areas, leaving the Adelie penguins out of the protection during the wintering period. We're also interested to see that how this area is used by other species during the winter, not only Adelie penguin. So when we consider the approximately the range of 80 until 100 degrees of eastern longitude, we can see that, for example, from a few other studies of wintering distribution of fulmar species, we can see that also they use the overlap of these three species is high, also with the area of wintering Adelie penguins. And as well, another example from the movements of juvenile emperor penguins, which also use the areas during this wintering period. For the coming steps that we would like to evaluate as well is the effect of sea ice conditions on the distribution and behavior of Adelie penguins, which is actually more ongoing than coming. And to conclude, in a nutshell, we found that the Adelie penguins from Ile de Petrel spend their wintering season in distant areas. The high interannual consistency that we found might imply the species vulnerability in the face of climate change, as it may indicate the low plasticity despite the variable environmental conditions. Also, we'd like to call to consider wintering period in the marine protected area proposal design, and also possibly adjust the Antarctic proposal to protect not only Adelie penguins in the critical winter period. And overall, we believe that our work would provide key knowledge to conservation and policymaking in the Southern Ocean. And thank you very much for your attention, and thanks all the funders for funding this research.
Thank you very much, Susanna, for your presentation. So we have time for questions. Yes, let's see if you have one. Yes. You have a question, okay, Susanna, from David. Okay. Interesting to learn about your work. Looks to me that petrel is ADPE are avoiding the wintry area of northern Victoria land, an ADPE, which are trying not to mix with the Ross Island, ADPE during winter. So we have a continuous belt of ADPE from Mary Bird Island across the Ross Sea to across Adelie land, each population trying to avoid the other. Would be great to have a collaborative paper among you, Anderson and Point Blue folks.
Yes, definitely it would be. Actually, we've been trying to do that, but yet we haven't got the funding. But if you have any idea where we could do that, I would be happy.
Yeah, the idea was to get funding to do a deployment all across the Antarctic among our different teams working on winter dispersal, to have a kind of synoptic view on 1 year. Sounds like a good idea, but apparently there's not enough money for that.
Yeah, exactly.
Okay, any more questions to Susanna? So if not, we still have like 2 minutes before finishing our session. Jose had raised a hand to ask something to Ryan. Jose, if you want to, we still have like 3 minutes. To finish
yeah, just very briefly. One thing is the excellent work that Ryan put into this paper has been amazing
and Jan and Mark Hindell on that work, which a later data set that Ekman put together. My question is just two questions. One is, are we definitely no problem having the scientific evidence where these hotspots are? It's clear for us from the scientific perspective. Would we need to say, oh, there's some gaps, we need to work on some areas still, like Jan just mentioned, that probably need to work on winter areas? Or are we comfortable so that we go back to our policymakers and say, particularly the ones that are more cautious on our work from the scientific perspective, go to them and say, from SCAR, from the scientific community, we are quite comfortable with this. We need to take action now.
Easy question, Jose.
Sorry.
Yeah, I think we have this discussion quite often. For me, I think there are I think the areas of ecological significance have a strong and robust basis. I think we can take those to policymakers comfortably. But I'm very curious to see the outcome of the KVA workshops, for example, to see in what ways that reinforces those areas of ecological significance. And there's a few other people also working on essentially the coherence among these different types of information. Sort of looking at the gaps, if you want to call it that, I think there are some. For me, always the South Pacific has been really interesting. So if you recall from the ratted work, it currently appears unimportant, and we need to figure out if that's simply a data gap in terms of our taxonomic coverage. For example, we didn't include some species which come from lower latitudes and feed in the Southern Ocean. It would be good to include non-central place foragers like cetaceans. So I think there are some spatial and taxonomic gaps that we do need to resolve to get an idea on those oceanic deserts, I think we called them in one of our workshops. But I think the AES are pretty solid. I think as soon as we have sort of confirmation from these other initiatives that are now ongoing, we can comfortably take those to policymakers and say, you know, these are places we need to conserve.
Thank you.
Okay, so I'd like to give the word to Michelle, because because this is the end of marine bubble session. So Michelle and Ryan did yesterday the talks, and I did today. So Michelle, if you have any words to finish on the session.
Yes, thank you so much, Manu, and fantastic job convening the session. Ryan, same to you yesterday. The last thing I guess I just want to say is thank you all the participants, attendees, speakers, and SCAR IT for facilitating a really great set of talks. I'm really excited about the work that's come out of here, and I'm already starting to see where some really nice new collaborations could possibly spring up. So I guess I'll depart by saying thank you again, and hopefully the next time we see each other in person, we'll be able to really advance some of the really great work that's been going on here. So thank you again, everybody, and have a great rest of your conference. Take care.
Thank you.
Thank you, Michelle, Jan,
and thank you very much, IT team. Namaste.
Ciao, ciao. Thank you.